So when we talk about making a genetic map, and again, this is just looking at relative different distances between one gene and another, we can do single crossovers, double crossovers, and you can do many, many more crossovers, but it gets super complicated, and so we won't do those. But looking at single crossovers first, and so we can see here from this diagram, so you have the tetrad, so it's during meiosis, and so you have, your maternal chromosome 2, say, so you have maternal chromosome 2, and say this is maternal chromosome 2, and then this is the copy of maternal chromosome 2, making those two sister chromatids. And then this might be paternal chromosome 2 and its copy, making those sister chromatids. And so that tetrad will come together, and then you have this crossing over event at a chiasma. And basically, these guys shown on the inside here will swap parts. And so you end up with these non-gamete crossovers. So this one here on the top, this one here on the bottom, right? There's no crossover event for them. So they stay the same, they stay parental. And so they are the non-crossover gametes or the non-crossover chromosomes, which then go to be gametes into the gamete. And then here in the center, you have these recombinant gametes or recombinant chromosomes that will then go into the gametes. And again, the reason we call it recombinant is because we had big A, big B, right? So the maternal chromosome here had big A, big B, and the paternal chromosome had little a, little b. No chromosome had a big with a little. And so that's why they're recombinant, because now there's a type of chromosome that didn't exist before crossing over. We have big A, little b, and little a, big B. So those crossover chromosomes then become crossover gametes. And those are the ones that are recombinant. And those are going to be interesting to us when we try to figure out how close these things are to each other or not, or how far they might be. And the sort of rule of thumb is, like we've discussed, is that the closer you are, the less recombination events you'll have, and the further away you are, the more recombination events you'll have, just because of basically real estate. If there's a lot of room between you, then a lot of opportunity for crossing over. If there's a little bit of room between you, then there's not a lot of room for crossing over, and less likely to have recombinant. So this diagram's a little bit busy, so I'll talk you through it. But basically what we're doing is cross A is comparing genes that are close together and therefore have low levels of recombination, and cross B is showing genes that are further apart, therefore having more recombination. That's basically the takeaway from this diagram. And so here at the top, you can see that you have the parental up here. And so we have the female and the male. If you're wondering about this sort of white hooky thing, that white hooky thing is just showing where a chromosome should be, but the genes that we're looking at, Y and W, are sex linked. And so the second chromosome there for this individual doesn't really matter. And so we kind of use this as a placeholder. It doesn't mean anything. It's just kind of weird for people to look at one chromosome when we just know there should be two. And so we put this placeholder there. There will be no crossing over between, the blue chromosome and the white chromosome, and they're not homologous, right? So anyways, just wanted to point that out. That's what's happening there. Is it's a placeholder. You don't need to really worry about it. Nothing's going on with it. It'll just be carried along as we go. So the parental strand here, the parental line here, you have YW. So on this side here, we have our YW double mutant homozygous. And then here we have YW homozygous wild type. Generally in our heads, we like to think about wild type as being dominant. I think it's a safe assumption for this course, but it's not a safe assumption in the world. So just remember that as you're moving forward in your genetics life, and should a problem specify that wild type is recessive, and that's okay. It's not a mistake. It does happen. But generally, you can assume that wild type is dominant for this course. And so here we have our F1 generation, and this diagram's maybe a little bit misleading. So this is the F1 progeny. So you can see it's a double heterozygote for both of those genes now, right? So a mutant and a wild type copy of each. This individual, however, is not necessarily from the F1 generation. What they are is a double homozygous recessive being brought in order for you to do the test cross. So this is the test cross, and the test cross is what's being demonstrated here by that X, right? So I can't put that there, but yeah. So that's what's going to be demonstrated by that X in the middle. That's a test cross. So we bring in a homozygous recessive individual for this as a test cross with the F1 progeny. And then you can see what the results are. And again, we separated males and females because this is a sex-linked gene that we're looking at, but we wouldn't always do that. Most of the data you'll see is just all lumped together. There's no delineation about males and females. It's just, here's your progeny with these different phenotypes. And so we can see that those genes are super close together, which means that their recombination frequency is very low at 0.5%. Okay. So that should make sense. There's not a lot of opportunity for crossing over to happen right between here. Whereas if we look at cross B, like, look at all the opportunity. I could make a million X's along here if I wanted to. Not really, because I'm using my mouse, not a pen. But anyway, I could do a whole bunch of X's. There's lots and lots of opportunity for crossing over to happen between W and M because there's just a lot of real estate there. There's a lot of chromosome in between them. And so what happens is, and actually there's 34.5 map units of chromosome between them, because they are so far apart that there's lots of opportunity for that crossing over event to happen. And in fact, it happens a lot more. 34.5% of the time in this experiment, we are seeing recombination. Okay. And so again, a recombinant is a version that was never seen before in the parentals. So the parentals were double. They either had, on the same chromosome, you had either the wild type of both, or on the same chromosome, you had the mutant of both, but you didn't have a mutant and a wild type. And so these recombinants have a mutant and a wild type. And so now we're going to see how we put these percentages into maps. And it's really easy. Once you have the percentages, you just change the units. So all you have to do is take that percentage, and then instead of percentage, you put map unit on the end of it, or centimorgans. So the example we're just looking at, so W and M, and we said that there was 34.5% recombination between them. Well, that's great, because then that means that there is 34.5 map units or centimorgans between them. And that's it. So once you can calculate the percentages, you just turn it into map units. So what do we have to do now? Well, we have to learn how to calculate those recombination frequencies or percentages. And we need to figure out how we figure out the order of these genes. Both of those things are pretty straightforward. So let's go ahead and practice them now. So we're going to start with just two. Okay, so just two, like maybe Y and W or W or M, just two. And then we'll come up, then we'll move on to these three genes. And that's where we're going to stop, because after that, it gets really crazy. So here's a practice problem. You're doing a genetics experiment with a fruit fly. In the parental generation, you cross two true breeding flies. So from your previous genetics life, you should know that true breeding means that you have only one type of allele. So whether it's the dominant or the recessive, if we're talking about Mendelian genetics, but you have only one type of allele to give to the next generation. The female parent is brown, so C, I guess, for color. So C plus, so brown, then we are saying is the wild type. And for this problem, we will assume it's the dominant. And wingless, W, so that's the mutant and the recessive. And the male parent is the opposite, C and W plus. And so if you want to do this problem using things like big C, little c, that would be okay. That kind of Punnett square notation will work for these types of problems. It just isn't right. Like, you're never going to read a genetics paper and see those types of notations. And so you may want to practice using the conventions with the pluses and lack of pluses, not minuses, but no, having nothing there. So the parental phenotype, or genotypes rather, they're true breeding, so that we know that mom is plus, C plus, C plus, and WW. The way I've written it here is one option. Another way you might see it is basically we write how it appears on the chromosome. And so you might see this, okay? So one of them sort of lumps the two alleles together. So we say two different chromosomes, two different alleles. This one sort of writes it how it is on one chromosome. So one chromosome has this, one chromosome has this. This notation makes more sense in the application in genetics, but at this point, I'm happy with whatever you're comfortable with. So this here is our female genotype. Won't let me back into it, but that's your female one. And so I'll just make another text box and we'll do the male. So the male is going to be sort of the opposite, CC, W plus, W plus. Technically, these are supposed to be superscripts, but I'm not going to waste the time with trying to fiddle around with superscript and things in the annotation tool. So we will just roll with this for now. So those, yeah, those should be superscripts. So C or so same thing. Okay. And so that would be the way that we would write it for the male and female. And so what happens when you have these true breeding parents together, whether, you know, the female is true breeding for both, like it has wild type both and mutant or mutant both traits, it doesn't matter. Like you're still, if you do the Punnett square on these two, you'll still end up with the double heterozygote that we like to see for these problems. So you'll end up with something like W plus C. Oops. W plus C plus WC, or again, you could write it as, I guess, W plus W, C plus C. Okay, so you can write it either way. Just get comfortable with one and be consistent. If you start mixing them up halfway through your problem, it's going to be complicated for you. Okay, so that would be our F1 generation, double heterozygous. So you have one wild type allele, you have one mutant allele for both traits. So now that we know that, that's sort of interesting, but not the most interesting part. What we want to do is go on to see if we test cross that F1. So you take an F1 female and cross her with a true breeding black wingless male. Okay, so black wingless male. If these genes are on separate chromosomes, what are the expected values for the genotype and phenotype ratio? And so I'm going to let you solve that one. That one should be straightforward. So that's sort of Mendelian genetics. If they're not linked, if there's independent assortment, what would you expect? But what we actually get are the counts below. So you get 85 brown winged, 728 black winged, 712 brown wingless, and 75 black wingless. So take a minute now and compare those back to the parents. Okay, so if you take a look back to the parents, and I guess I could scroll back too. So the parents were, wow, brown and wingless, and black and winged or normal wings. And so you can see that these ones here, nope, that's not a highlighting tool at all. So these ones in the center, the 728 and the 712, are the parental types. And that's really noteworthy because recombination will happen and you can be, you know, up to 50 map units or 50% recombination is sort of as far away as you can get between the two genes. And so these ones in the middle here, these are going to be your parentals. And the parentals will always have the biggest numbers. And that's just sort of because how recombination occurs and where that chiasma can occur. You know, we're always just looking at 2 or 3 genes, but of course there's tons of genes down the whole length and the chiasmatic can occur anywhere. But the parental ones, those sort of ones on the outside that we looked at, we look at the tetrad, more often than, you know, those ones are going to stay the way they are more than the ones that are changing in the middle for those genes. And so we can see here that these middle ones are our parentals. So that's great. So our recombinant ones then are these 85 and these 75. So that's cool. We can figure that out. And so what we want to do is find the recombination frequency of the percent recombination. And so what we do is very simply with these, you know, sort of 2 gene crosses, you add up your recombinants and you just put them over the total. And spoiler alert, if you add all those up, you get 1600. And so if I add up my recombinants and put it over the total, you end up with 10%. Okay, so 10% recombinant. Therefore, 10 map units or, whoops, 10 map units or 10 centimorgans apart. So that's interesting, right? So if you can do, so you do your recombinants, maybe I'll just write that down here, up here, sorry. So you do your recombinant, add sum of recombinants over total progeny. Okay, so you do your sum of your confidence over your total prodigy times 100 because it's percentage. And then you turn that percentage into map units and centimorgans. And again, you can use those interchangeably. So that's pretty straightforward. So those genes based on our experiment are 10 centimorgans or map units apart. Awesome. So that's kind of, yeah, that's exactly what you would do with those. And sometimes, you know, that's about as complicated as those questions get, is, you know, looking at two genes isn't super exciting. But sometimes we can throw in a third gene, and that's where we're going to head. So let's just take a look at including a third gene and what we can do with that. So with the same set of data, we have this wing gene, so the W plus, and then we had this color gene. And we know that the wing and color are 10 apart. So let's just bring that information forward. So we know that C and I'm going to use C and W are 10 map units apart. Okay, now we have some new information here that says that antennae length is 5% recombination when you look at W. Okay, so between W and antenna length, which I don't know, we can call whatever we like. I guess we could call it just A. Just call it A. So antenna length is 5%. So list all possible recombination frequencies between the gene for color and the gene for antenna length. So the first thing we want to do is draw this out. And drawing this out would be a totally appropriate answer. So we know that C and W are 10 units apart. So you might on this put C, a little bit hard with the annotation tool, W, and then we know that antenna is 5 from that, right? So maybe this is how they sit. And so if you were drawing that, you might write in what the distances are. So from here, oh, this is going to get sketchy with my mouse pad, right? So this one here versus this, oh yeah, see, there it is, ugly. So we might just put in those distances. Ah. And so we would say this one here is 15. So the possible recombination frequency between C and A would be 15. Between C and W, know is 10. And between W and A, we know from the problem above is 5. So that's one possible permutation of this. The other option is it could go C, and these are not to scale by any means. I can't hold myself to that. CAW, that was not bad, I guess. So it could also go CAW, right? Because all I know is that A and W have to be 5 apart and C and W have to be 10 apart. I don't know anything about C and A. In fact, that's what I'm trying to propose. And so in this instance, I might write an annotation where I say, okay, C and A, whoop, oh, ugly again, apologize, and A to W, and then C to W. And so you just want to make it very clear that you know that this is additive and that what the actual recombination frequencies are. And so from C to W, we said was 10. And we could propose that another recombination frequency between C and A would be 5. So list all possible recombination frequencies between C and A. Given a linear chromosome, of course, if it was circular, there'd be a whole other mess. You know, you can keep going. But you would be between C and A, you could have 15%, or you could have 5%, depending on where A is relative to C. We know that it's 5% away, or 5 map units away from W. So this would be what you would submit. You'd submit these little maps. They're not too fancy, with some indication of you knowing what numbers and indicating how the order impacts the numbers. And that's it. That is what happens. That's how we process from 2 genes, looking at C and W initially, adding in a third. Now, in this case, the third one, we gave you the percentage, which is really lovely and generous. That is just not how it's going to be. You're going to have to calculate the percentages between all 3 of these genes. And that's where we're going to head with our 3-point maps. And so that'll be the next video.